Greetings and welcome to the Tonix Pharmaceuticals Planet MicroCap Webcast. It is now my pleasure to introduce your host, CEO Seth Lederman. Thank you, Mr. Lederman. You may begin.
Thank you very much for the introduction, and thank everyone who's participating in watching the webcast and sending in Q&A for your interest in Tonix and our story. I'm Seth Lederman, CEO of Tonix, and I'm going to give you an update on our story today. This is our standard safe harbor disclosure, and we will be making forward-looking statements. We're a publicly listed company, and I refer you to our 10-K for more complete information about the company. These are our two lead programs on slide 3. The first, with a lot of interest already in the Q&A room, is our vaccine to protect against COVID-19. This is in the preclinical stage of development. It's a live virus vaccine, and it's designed on our Horsepox vaccine platform to express the CoV-2 spike protein. And we will be having animal data this in the next quarter, so third quarter. And I'll tell you more about it as we go through. Our other lead program is TNX-102SL for fibromyalgia. That's in phase 3 development, and there's a phase 3 study called the RELIEF study that's currently enrolling. The fibromyalgia program is based on sublingual, that means under the tongue, cyclobenzaprine tablets. And we will be having a lot of data next quarter. The interim results of the phase 3 study are expected, and we expect to have top-line data in the first half of 2021. Because of the interest in the COVID vaccine, I'm going to spend most of today talking about the COVID vaccine, but that's just because of the interest in it, not any statement about our excitement about the fibromyalgia phase 3 study. On slide 4 is it shows the 3 vaccines that we have in development that are all based on or related to the Horsepox program. So I just want to introduce these vaccines to you so you can understand how we got here. The top one, TNX-1800, is the vaccine that we're developing to protect against COVID-19. And as I said, this is based on our Horsepox virus, and it's expressing a COVID-19 protein. The next on the list is TNX-801, which is a vaccine against smallpox and monkeypox. And I'm going to spend some time talking about TNX-801 because that's really how we got to the COVID vaccine. And TNX-1200, you'll see that mentioned in the slide later in the deck, but that's really, from the point of view of this presentation, a control vaccine based on an old-fashioned Vaccinia virus, which is the traditional vaccine for smallpox. Slide 6 takes us back to 1798. One of the most famous books in the history of medicine is Edward Jenner's Inquiry. And this is the discovery and description of the first vaccine ever. And that vaccine was a vaccine against smallpox, and Edward Jenner called it Vaccinia. So that's why we still use the term Vaccinia today. But these are 2 quotes on slide 6 are from the 2nd and 3rd page of the inquiry. And I'm showing them here with the words horse underlined to emphasize that Edward Jenner got his first vaccine from a horse. He believes that it was communicated from the horse to a cow by the unwashed hands of the people who would put the hooves on the horses. They're called farriers. So the idea that for us to work on Horsepox was really to go back to, as close as possible, the vaccine that Edward Jenner was working with in 1798. And this is a very modern analysis of Horsepox, which is shown at the very top in a blue line. And below it are the sequences of a number of Vaccinia viruses, and at the very bottom, a different virus. But without taking you through too much molecular biology, the Horsepox virus is very closely related to Vaccinia viruses. And the big difference is that the Vaccinia viruses are missing chunks of DNA on the left side and the right side. So if you see in the top part of the diagram, the chunks missing on the left, the white space is pieces of DNA missing from Vaccinia, and on the right side, also pieces missing from Vaccinia. One of the other interesting things about this is at the very bottom of this stack is Variola, and that's the virus that causes smallpox. You can see that Variola also shares a lot of the deletions, particularly on the left side, that the Vaccinias have. And the fact that Horsepox has more DNA in it, we believe, and others believe, means that it is related to a more primordial virus, and we think related to the virus that Edward Jenner was working with. This slide 8, shows an experiment that we did with our Horsepox vaccine in a model for smallpox to protect against smallpox. And since no one does research, it's not ethical to use Variola because it's such a terrible human pathogen, that the model that's accepted is to use monkeypox, which is abbreviated MPXV. And I'll come back to the first two panels, but maybe if you look all the way on the right, this is what happens when animals are not vaccinated. They're given a sham vaccination. And in this case, 4 out of 4 macaque monkeys got sick and had lesions on their bodies. And the graphs, the lines that you see, the 2 blue lines and the 2 red lines, are the lesions, the number of lesions on the body of basically unvaccinated monkeys. And the reason why 2 of them are short is monkeypox is a lethal virus, so unfortunately, 2 of the animals, had to be euthanized. But on the panels on the far left, on the 2 left, show what happened when we immunized first with our TNX-801 horsepox virus. And as you can see, there were no lesions in any of 8 animals. And I'll also mention that all of the animals were healthy and they didn't develop any illness from the challenge with monkeypox. Then in panel C is a control where we vaccinated with something that is similar to a traditional Vaccinia vaccine. You can see while the animals were healthy, 2 out of 3 developed lesions. So the protection that we saw with Horsepox was very impressive and strongly motivates us to pursue Horsepox as a vaccine for smallpox and monkeypox. On slide 9, there's a description of TNX-801. As I mentioned, it's a vaccine based on Horsepox. This Horsepox clone that we're working with was synthesized, but it doesn't contain any new gene elements relative to the environmental isolate. We think it's a primordial strain of Vaccinia, since the left and the right DNA sections that I pointed out are complete, whereas the modern Vaccinias have deleted them. There's several other attributes that I don't have time to go to, but it has a small plaque size in culture, which is similar to the environmental horsepox. It has substantially decreased virulence in mice, and that has been published. And as I just showed you, it protects monkeys from monkeypox. And we believe that the molecular evidence showing that this is closer to the vaccine that Edward Jenner and others were working with between 100 and 200 years ago indicates that this has been used in humans with extensive experience. And horsepox, although horsepox hasn't been reported in more than 40 years, we think the fact that it has basically not been absorbed for 40 years is a sign that it's relatively safe from the point of view that with good animal husbandry, basically hand washing and other techniques like that, it really hasn't been a problem in the horse in horses in modern times. So slide 10 shows it introduces the idea that horsepox is not only a vaccine potentially for smallpox and monkeypox, but we believe it's a vector platform for other infectious diseases. So horsepox can be engineered to express foreign genes and to serve as a platform for vaccines. Horsepox has a large packaging capacity for DNA inserts. And, as I mentioned before, we're putting a DNA insert into horsepox to express the CoV2 spike protein. In addition, horsepox has the advantage of precise virus-specific control of expression of these inserts. Very importantly, there's a lack of persistence or genomic integration in the host, and this is a key safety element of viral vector systems. It has strong immunogenicity as a vaccine. That's an important criteria. As I'll get into later, we really want to engender T-cell immunity. And we believe that horsepox is strong in that capacity. It has the ability to have new constructs made quickly to respond to a new virus like the current pandemic. It's manufacturable at scale, which is important because, as I think everyone on the phone knows, the potential of a COVID vaccine would mean that hundreds of millions, potentially billions of people would potentially be vaccinated with it. And finally, it's a live replicating vaccine, which means that it presents antigens to the immune system in a way called direct antigen presentation, which is a very powerful way to turn on T cells. Now, I've also mentioned that horsepox is very similar to Vaccinia, but it's similar, but we think it has advantages. First, the advantage is that we think it has improved tolerability. We've shown that in mice. We had good tolerability in the monkeys, but it wasn't done in a way to compare it with traditional Vaccinia. But, its small plaque size and other characteristics lead us to believe it may have improved tolerability. I've mentioned before that we have direct antigen presentation, and there are types of non-replicating Vaccinia which are being advocated for their safety, but we believe they don't have as strong T-cell stimulating properties as horsepox. And finally, at the bottom, horsepox may depend differently as a vector because of a whole different repertoire of genes that modulate the immune responses and host range. And these genes are found on the left and the right side where there have been deletions in Vaccinia. So in the next slide, slide 11, we show the vaccine that we're developing against COVID-19. At the top in blue is a picture of the horsepox genome. And below it, in flesh color, is an expanded view of a gene that we just called gene B. In the middle, where it says homologous recombination, that refers to a process in which one piece of DNA can be substituted for another piece of DNA in the Horsepox genome. And at the very bottom of the slide, where it says TNX 1800, that shows the new vaccine that is designed to express the SARS-CoV-2 spike gene. So TNX-102SL is a recombinant virus that expresses the CoV-2 spike gene with the idea that immunizing people with TNX-1800 will result in immunity to the spike protein. On slide 12, we describe some of the advantages of Horsepox as a vector platform for a SARS-CoV-2 vaccine. And these are points that are specific to the biology of CoV-2. First, Horsepox delivers strong immunogenicity for adaptive and innate immunity. And both of those arms of the immune system are believed important in SARS. Underneath are some smaller points, and I won't go through all of them, but I will highlight that T-cell protection is very important, we believe, for protecting against COVID-19. There hasn't been much laboratory work done on COVID-19, but a lot was learned from SARS, which was the coronavirus that infected many people in Asia in 2003. And in those cases, it was shown that a T-cell response to the spike protein was important and was durable for over a year after SARS. There is a lot of other biology from the SARS experience that points to the use of Horsepox as a vector for SARS antigens to protect against COVID-19. In the middle point, we talk about our collaboration with Southern Research. Southern Research is a nonprofit that is very well regarded for the work they do on infectious agents. And they are set up to study the CoV-2 virus and to study it in animal models, including animal models where that virus or related viruses are used to infect mice or non-human primates. And this is a way to study the effect of a vaccine. And we are collaborating with Southern Research, and that is where our experiments on mice and non-human primates will be. And as we've indicated, we expect to have data from these experiments next quarter, so in the third quarter of this year. And finally, at the bottom, further development. A big focus of Tonix right now is manufacturing TNX 1800 for human clinical trials according to good manufacturing practice, or GMP standard. So we're very actively engaged in beginning the manufacturing development that will enable human clinical trials. Well, now I'm going to change from our COVID vaccine program, and in the last few minutes, tell you about our pipeline. So on slide 13 is a slide showing our candidates in development for pain, psychiatry, and addiction. And as you'll see at the top, TNX 102SL as a bedtime treatment for fibromyalgia is in a phase 3 study that is currently enrolling. And we are expecting interim results in the third quarter of this year, so next quarter. And that is a phase 3 trial. Fibromyalgia is a pain condition. It affects people, mostly women, and the average age of onset is about 50 years old. So we're very excited about this program, and it's our most advanced program in development. Also in our pain, psychiatry, and addiction portfolio is TNX 1300, which is a treatment for cocaine intoxication. And that's a biologic medicine, and it has FDA breakthrough therapy designation. We licensed this from Columbia University, and it is in it's currently in this phase 2 stage of development, and we're planning our next phase 2 study. And finally, among the programs I'll talk to today, TNX 601, controlled release, which is a potential new treatment for major depressive disorder. And we expect that to be in the clinic in 2021 or next year. And now, in the interest of time, I'm just going to advance to the to slide 36. And slide 36 is a roll-up of the milestones that you can expect to hear from us. This quarter, we expect to file the IND, or Investigational New Drug application, for TNX 102SL to support a phase 2 study in alcohol use disorder. Alcohol use disorder is the modern term for alcoholism. And then in the third quarter of this year, you can see that we have a number of major events. First, in the third quarter, we expect to get small animal data from our COVID-19 vaccine, TNX 1800. Also in the third quarter, we're expecting primate data from the vaccine program. And then also in the third quarter, we're expecting interim analysis results from our phase 3 fibromyalgia study. So that's the end of my prepared remarks, but I do want to answer or respond to some of the questions that have been put onto the Q&A tab from you in the audience. And I don't have time to respond to all of them, but I can see that most of the questions are about our COVID-19 vaccine program, asking about timing for when people can expect data from us. And I think the milestone slide directly addresses those questions. As is said on the milestone slide on slide 36, small animal data from the TNX 1800 program is expected next quarter in Q3. The primate data from the COVID-19 TNX 1800 vaccine program is also expected in third quarter or next quarter. And that's the same quarter when we'll be expecting the interim results for the phase 3 fibromyalgia study. So with that, I thank you all for your attention, and now I'll turn it back to the speaker organizers.
Thank you so much. This does conclude today's webcast. You may disconnect your lines at this time. Thank you for your participation.